Hello everyone, I'm Shengbo Wu from Zhejiang Institute of Tianjin University. Today I'm very happy to attend IFHN 2026. My report topic is mining and validating quorum sensing interference molecules from food-derived compounds for Salmonella Typhimurium. Today I will introduce four aspects. Firstly is personal profile. I obtained my bachelor, master, and doctor in Tianjin University. Also, I'm a visiting scholar in University of Oxford. For now, I'm an associate professor. My research interests including quorum sensing, synthetic biology, food microbiology, et al. And my research integrates multidisciplinary theories and technologies from bioinformatics, systems engineering, and synthetic biology, aiming to systematically decipher and modulate gut microbiota for improving nutrition and human health. I have published over 20 research articles in journals, including Nature Communications, Trends in Microbiology. My research interests including three aspects: AI-enabled QS discovery and design of synthetic microbial consortia, and food microbe and host communication. And then I will introduce some research backgrounds. Firstly, quorum sensing is a key molecular mechanism in microbial networks, influencing many psychological activities, such as biofilm formation, drug resistance, virulence, and some other phenotypes. And for example, quorum sensing can be used to coordinate fluorescence expression, communication, and cooperation with the host, and even monitor host immunity. And quorum sensing has different systems such as AHL, CI1, DSF, and AI3, AI2. Salmonella infection of the host consists of 5 steps, including adhesion, invasion, propagation, spread, and symptomatic infection. QS-based regulation of Salmonella depends on different quorum systems, including AHLs, AI2, and AI3, and Indo and DSFs, which are interconnected and influence each other. And quorum sensing molecules which have been detected in samples from the human gut system. Mammalian receptors present in the human gut may be able to bind the different quorum sensing molecules, such as the AHR and some T2R and some other receptors. Also, quorum sensing have been reported to attend or involved in different food systems. As for the left part, many different quorum sensing systems can be attended in the spoilage or pathogenesis of the different pathogenic bacteria in food systems. Also, in the right hand, quorum sensing can be also helpful for the food system, such as the biotransformation. And recently, machine learning empowers microbial resources mining as well as the question and obtaining some bioactive small molecules. There are some effective tools have been developed in recently, such as the EVO2 and the largest publicly available AI biological model, and also its EZ specificity, the high precision prediction of enzyme substrate specificity. Also, the deep sea maps, which is used to reveal the hidden biosynthetic potential of the global ocean microbiome with a large language model. And in the last decade, we have developed an AI-based platform for QS mining and analysis. As for the databases, we have developed the QS molecules, QS genes, and QS proteins to develop a series of databases such as QSIDB, QSHGM, QSDMD, and QSCHGM. Also, we have also developed 2 AI classifiers. Number one is the QSPRED, which is binary classification based on feature extraction AI organisms. Also, the QSDeep is a multi-label classifier based on AI language model. And finally, we have constructed a knowledge map, which including 9 languages and 36 phenotype regulations. As for the academic impact, we are very lucky to be invited by Springer to contribute chapters to 2 English language books. Also, the senior QS researchers group of Sharia commented that the QSHGM is an exciting tool for users to explore, and many of the series work have been cited and praised by genomics teams, including the different teams from all around the world. Thirdly, I will introduce the academic achievements. 2 studies based on the food, quorum sensing, and microbe interactions. Number 1 is the published work in food function in 20 and 25, mining and validating quorum sensing interference molecules from food-derived compounds for Salmonella. Firstly, is the virtual screening-based calculation and analysis of the potential QSIMs. In this work, firstly, we used the autodoc winner to calculate the binding of different food-derived compounds and the 9 quorum sensing receptors. Results show that most food-derived molecules bound widely to the QS receptors, but rarely to LSRB, which indicates the strong specificity of the LSRB. So we further to screening the different food-derived compounds for LSRB to mine the QSIM interfering biofilm and virulence. The second is the LSRB-based mining of the potential QSIMs. In this part, we find that the 6 food-derived molecules will bind the Salmonella LSRB, and the one is scheduled to aminoglycine and typhimine, and 3 phenylactic acids, and the other 2 compounds. And then we used the technology of SPR and PIMO to confirm the specific hydrogen bonds and the pi-pi interactions with key LSRB residues. The third is the performances of QSIMs on the biofilm and motility. In this part, we further find the schedule to animal quinoline and typamine. It will inhibit the Salmonella Typhimurium biofilm formation and thickness. Also, the 3 phenylactic acids and tricarbalic acid and L3 phenylactic acid will promote the biofilm growth and thickness. Finally, we find the typamine 3 phenylactic acids and L3 phenylactic acid will promote both swarming and swimming motility. And then we will validate the QSIMs at the molecular level. We conduct the QRT-PCR to investigate the following genes which will be influenced by different QS systems. We find that the schedule and 2 quinoline will inhibit LSRB, FLHC, and AI2 pathway. The typamine does not will affect the LSRB. And the 3 phenylactic acid and L3 phenylactic acid will promote LSRB, FILA, and the AI2 pathway. And except the typamine and 5 QSIMs will model the AI2 pathway by targeting the LSRB receptor, which is constant with the results of QRT-PCR. And also, we further validate the QSIM at the human cellular level. In this level, we conducted the different QSIMs to the Salmonella infection, namely is the invasion and adhesion of the human cellular human cells. We found that The schedule and annual quinoline will inhibit Salmonella Typhimurium adhesion and evasion to host cells, and the tricarbalic acid and L3-penylactic acid will promote adhesion and evasion, just as the QS agonists. And then I will introduce the work which is not published in the for now, and maybe it is will be accepted in some other journals, named as Discovery of Dietary Polyphenols Targeting the AI2 Quorum Sensing Systems of Pathogenic Bacteria. First part is training and performance evaluation for AI2 QSIM classifier. In this work, we established a machine learning-driven systematic framework to identify different pathochemicals that will interfere with AI2 pathway across bacterial pathogens and elucidate their mechanisms. And this framework, including the training, screening, testing, and validating model. And the right figure shows that the AI2 IM-PRED GN classifier has a good performance for classifying the molecular, which reveals that the natural compounds constitute the only 19% of no AI2 QSIMs. So it is essential to mine the more QSIMs based on AI2 pathways. And the second part is the screening polyphenols QSIMs based on AI2 IM-PRED and LSR biosensor. In this part, we find that polyphenols will dominate AI2 QSIM candidates. 38, and 263 show high antibacterial activity, and 26 polyphenols will moderate LSR operin expression. Among them, 21 will suppress the activity, and five of them will enhance the LSR operin expression. And we conduct the receptor testing of polyphenol QSIMs with the AI2 pathway. The AI2 pathway, including the synthesis based on LuxS and the transportation based on TQSA and the interaction with the TF, LSRR, and this is the circle. We find that the 8 polyphenols will bind LuxS or LSRB with the efficient KD values. And with specifically, we find the proteosprinine will target LuxS, and for methyllactol, pyroglycerol, and 3-methyllactol will target LSRB receptor. And then to further characterize the common scaffold of LSRB-bound polyphenols, we do some expansion. We expanded the molecules types as three classes. The class 1 is the polyphenols will substitute only hydrol. This class 2 is substitute with the hydrol and lower hydrocarburyl groups such as mercury, ethyl, and propyl. Class 3 is the polyphenol will be substituted along with methox and ethox groups. And the results showed that the class 2 polyphenols will show stronger SRB binding than class 1 or 3, especially with para substitution. And all 3 classes will suppress AI2 quorum sensing pathways. Class 2 disrupt the system mostly efficiently by forming more hydrogen bonds with SRB. And then to validate the polyphenols QSIM, we use the different experimental levels, including genetic, strain, and cellular assays. Other experiments results showed that the 10 polyphenols will target LAX-S or LSRB, down-regulating Flagella, SP1, SPI1, and LSR operon genes. And polyphenols will inhibit motility and biofilm, will also enhance antibiotic sensitivity and reduce host cell invasion. Finally, we find genetic, strain, and cellular level validations will confirm the polyphenols interfering with AI2 pathway in Salmonella Typhimurium. And finally, we propose the universal mechanism by which distinct polyphenols will interact with various AI2 effector proteins, such as LAX-S, LSRB, TQSA, and LSRR, to modulate quorum sensing-related phenotypes. The different responses of the pathogenic bacteria to polyphenols are likely due to the variation in completeness and conservation of AI2 effector proteins. And finally, I will introduce the summary and acknowledgement for these two studies. Machine learning will access the mining of quorum sensing interference molecules to inhibit pathogens and alleviating drug resistance. Polyphenols will inhibit biofilm formation, motility, virulence, gene expression, and host cell invasion across multiple pathogens with AI2 systems. The food-derived QSIMs will offer a promising diary strategy to weaken salmonella infection, while the food-derived QSIMs can be used to combat drug resistance and guide broad-spectrum antivirus therapies. And polyphenol-mediated diary intervention can modulate gut microbiome composition and abundance. Well, the polyphenol-based QSIMs can offer a novel strategy for preventing and treating microbiome-associated diseases. So finally, thank you for all the collaborators from Netherlands, the Professor Jingyuan Fu and the Professor Jianjun Qiao from Tianjin University, and the Fei Guo from Central South University, and Professor Ai Dongyang from University of Oxford. Finally, I will also thank you for the fundings from China.